today I had a talk about molecular genetics and its importance for diagnostics of thyroid cancer. I hope that at all types of the mutations which are typical for thyroid cancer. So it's a well-known fact that cancer is induced by different mutations, and though different expression profiling and methylation changes are very important for development of cancer, still we believe that the primary event is the mutation. And so we are looking if we can establish the new diagnostic technology based on detecting all known mutations which induce different types of thyroid cancer. To the date, there are several approaches. So first of all, we may look at the so-called hotspots or known mutations which are really frequent in this or that type of cancer, and it's possible to establish rather simple molecular genetic technology based on common polymerase chain reaction PCR, and the only limit is that you're looking only to this particular known mutation, and very often it's not enough because you have no mere mutations, you have a lot of rare individual mutations, or even each time de novo mutations, sporadic mutations in different patients, and you cannot cover with the testing the known mutation all the spectrum of the mutations. So the second part is looking at the chromosome aberrations, which is very typical for many cancers, and for example, we are also working with chronic myeloid leukemia in our laboratory, and this is a very beautiful example of the cancer which is induced by the chromosomal translocation in all types of this cancer. So this is a very unique situation when you have the single mutation which is very typical for this kind of cancer. You can find this mutation and put the diagnosis, and vice versa. But in general, translocations are very frequent in different types of cancer, and in most of them they happen sometimes, and sometimes other mutations happen. So in thyroid cancer, chromosomal aberrations are rather typical, and we are also going to check for them. And the third one is quite new approach for looking the copy number variation mutations. This is a case when you have the loss of the mutation, the loss of the gene of the allele, or vice versa, multiplication or amplification of the allele of the gene. And this is why it is quite new, because we were capable of detecting such kind of mutations at large scale when we were working at chromosomal level, looking in microscopes. But there is a limit of several megabases at the best, or over 10 megabases, so million nucleotides. And very often it was shown that very often copy number variations, they happen in smaller size, like thousands or sometimes hundreds of nucleotides. And it is impossible to look at the whole genome for this kind of mutations. We have to upgrade our technologies. And finally, we got this upgrade with next-gen sequencing, when we are capable of looking at the whole genome if we want. But for this particular case, it's enough for us to look at about, let's say, several hundreds of thousands of nucleotides, the most common mutations of this type in thyroid cancer. So we are trying to look at several hundred of hotspot mutations, or single nucleotide mutations, at something like 2 or 3 dozens of chromosome translocations, the so-called fusion genes, generating fusion genes. And the third one is copy number variation, about 3 of them. All of them, they are based on technology, from the point of view of technology, they are based on doing next-gen sequencing. So all of them are based on the same approach. I would say that it's about 500 mutations, including all of them, all of which I counted. But finally, it's unknown. Maybe we will, I mean, not we, but we as humans will find more, like thousands. But I believe that in thyroid cancer, we will stop at about several hundreds, maybe up to 1,000. Yes, that's a big problem of big data, and hopefully for this particular project we are not generating a lot, because the region of the coverage is relatively small compared to the other approaches and other projects with next generation sequencing, so like exome sequencing, which we performed for chronic myeloid leukemia. But yes, that's a problem. So the only thing which is not the problem in this project for a while, because we are just gathering the data, clinical samples, we are just beginning. We designed the panel, we've got just few first samples. So I wouldn't say that I'm ready to share some of the results, for example, not yet. For now, we are looking at the hereditary basis of mental retardation, for example. But if to come back to thyroid cancer, so most of the mutations, they are sporadic mutations. This is not the hereditary case. Though there are some hereditary types of thyroid cancer also, and I believe that we cover at least the most well-known mutations which induce hereditary or familial thyroid cancer. But these cases usually are not so important for molecular genetic diagnostics primarily. So finally you have to confirm, of course, but primarily you get enough clinical data to establish a hereditary feature of this disease, because you have the family. It's a very hot topic. Everyone is talking about genome editing. Everyone hopes to cure all hereditary diseases and most of infectious diseases using this technology. And we focused on one of the most frequent monogenic diseases, cystic fibrosis, which is also included in Russia in the neonatal screening program. So because it is one of the frequent and curable diseases, well, not curable, but treatment is available for this disease. And there is a major mutation, the deletion of 3 nucleotides in this gene, and that's why it's possible to focus on this major and the most severe type of the mutation. So we are trying to cut the gene in this place and to replace the deletion with the normal sequence. So we are not the alone, and it's a very hard, competition, because a lot of companies are working with CRISPR technology, and they are also focused exactly on the same disease or other diseases from the most frequent ones. And yeah, it's very interesting and very, very, emotional. To the moment, the only available working diagnostics for thyroid cancer is cytologic and pathomorphologic analysis of aspiration biopsy, of aspiration biopsy samples. And the question is that this approach needs very highly educated and highly trained specialists who are able to distinguish between normal and cancer, which we had the opportunity at this session to see that it's really difficult to distinguish if it is normal or cancer in many of the cases. And this results in over and hyperdiagnostics in about 20–30% of cases. So it depends greatly on who is doing the research and who is performing the diagnostics itself. So in some cases, people claim that they have the mistakes in only a few %. But mainly meta-analysis, they show that the mistake is about 20–30%. And we see these patients come to these patients, so the samples of these patients come to us for molecular genetic diagnostics, but when they come to us in a hurry because it's an operation and we have to take the fresh material quickly, and this is difficult to perform, and finally we get the sample with diagnosis benign, because first diagnosis was likely tumor, likely cancer, and huge operation like thyroidectomy or hemithyroidectomy, and the intraoperational diagnosis benign. It's a big problem for patients, of course, and we also have the vice versa situation when benign on the preliminary diagnosis and no treatment and progression of the tumor. So we believe that with our technology of molecular genetic diagnostics, we will be capable of diagnosing over 95, or in upper limit it should be 98.6%, as it follows from the investigations of the so-called TCGA. This is the consortium working with different cancers, and they demonstrated that if we gather all cancer samples of thyroid cancer, we will find these types of mutations, single variant mutations, copy number variations, and chromosome aberrations in about 98.6% of all the samples. So we believe that if we are looking at all of the mutations, we will find 98 correctly. So this is our contribution to diagnostics and hopefully to correct treatment. It's possible to add to the panel, and I think we now maybe lack these mutations, but it's easy to add them to the panel, the mutations which help to choose in some of the cases targeted therapy for the non-mutations which can be treated by specific drug. So it also works, can be used.